The MapMed project is a multidisciplinary project founded by HEMPI, Cross-Border Cooperation in the Mediterranean, aimed at improving the environmental sustainability of tourist ports with regard to marine pollution and with a specific attention to petroleum hydrocarbons. Ports are critical environments since they are located between sea and land, and they receive pollution both from ships, from land, and also from port facilities. Moreover, they are not closed systems, since pollution from the ports can impact surrounding coastal areas. Particularly, tourist ports are subjective to a seasonal anthropogenic impacts, which increase during the summer season. In this slide, I show you the pollutants entering tourist port by anthropogenic impacts, which are organic matter and nutrient factors deriving mainly from sewage water, as well as petroleum hydrocarbon from marine traffic and port facilities. In the Water Framework Directive, ports and also tourist ports are considered as heavily modified water bodies, since they have strong modification of the hydromorphological properties. The final goal of our project is to provide port authorities of tourist ports with tools for the implementation of an environmental management system to guarantee the sustainable management of our tourist port areas in the Mediterranean Sea. Thus, the environmental management system is aiming to improve the environmental quality of ports by understanding and monitoring the evolution of the quality of the system and defining a set of measures for bioremediation of waters. In order to achieve this final goal, the MapMed project has been divided into three phases, which are the site characterization, eco-efficient water management and bioremediation, and support to institutions. For the implementation of the project, 4 case study sites has been selected, 2 located in the European country, Cagliari in Italy, and Heraklion in Crete in Greece, and 2 in the Mediterranean coast, in the South Mediterranean coast, Il Kantawi in South Mediterranean coast in Tunisia, and Porto Marina in Egypt. The final goal of the site characterization is the definition of a knowledge framework of the 4 selected ports with regard not only to geographical but also chemical, physical, and ecological properties. And these slides show you the strategy that we use. So we choose an integrated number of parameters, chemical, physical, but also biological, from bacterioplankton, but also zooplankton and phytozooplankton, but also seagrasses, and of course, hydrocarbon measurements and hydrocarbon degradation. And here I show you results of the comparison of the 3 ports that we analyzed during 2012. We performed 3 different sampling campaigns in winter, in spring at the beginning of the tourist season, and in late summer at the end of the tourist season. Unfortunately, in 2012, due to the unstable situation, we were not able to collect samples from the Egyptian ports. So I compare here data of the other 3 ports. And in these slides, you see inorganic nutrients, which are nitrogen and phosphorus level, and POC, which is particulate organic carbons, and the statistical analysis of all the data demonstrates that Cagliari port has significantly higher concentration of nitrogen and phosphorus, but also particulate organic carbon with respect to the other 2 ports. Here are the viable titles of heterotrophic bacteria, and you see here that also title of heterotrophs is significantly higher in Cagliari port as compared to Heraklion and El Kantawi port. These slides show you the level of hydrocarbon in the 3 pores, and you see polycyclic aromatic hydrocarbons are not significantly different between the 3 pores analyzed, and on the contrary, aliphatic hydrocarbons showed significantly lower concentration in Heraklion port as compared to the other 2 pores, while the mean value of aliphatic hydrocarbons is higher in the Il-Kantawi port. And it's interesting to see here that exactly the same situation have been observed as regard to diesel degraders, since we have a significantly higher, slower concentration of degraders in Heraklion pores as compared to the other 2 pores. Then we analyzed the degraders. We perform 16S analysis, of course, for phylogeny. And it's interesting to know that in accordance with the trophic level of the 3 pores, isolates from Cagliari were assigned to genera of copiotrophic bacteria, mainly Pseudomonas, but also Mycobacterium, while isolates from Il-Kantawi and the Heraklion pores belong to genera of obligate oligotrophic hydrocarbon degraders, mainly Alcanivorax and Marinobacter. So we arrived to the second phase of our project, that is the eco-efficient management and bioremediation. Here we decide to choose suitable bioindicators for monitoring of water quality and bioremediation. And we selected a subset of parameters based on the site characterization, and we decided to test these parameters in the Il-Kantawi port, in the Tunisian port. Il-Kantawi port is a small marina located in the coast of west coast or east coast of Tunisia. It's located in a small village without permanent inhabitants, so tourist activity is the only anthropic pressure on this system, and this is one of the reasons why we chosen this system for our trial, field trial. Moreover, as I show you, this small sport had the higher concentration of aliphatic hydrocarbons in seawater. In accordance with the Water Framework Directive, we divided the basin into two management units. One management unit where docking of leisure boats is located, and the second one where you have the infrastructure from the port facilities, thus fuel station and shipyards. And we selected the 4 stations in the leisure boat sectors and the fuel, and 2 in the unit 2 near the fuel station and the port entrance. Then we perform a bi-monthly monitoring, and here in the table you see the monitoring parameter that we follow, so chemical, physical, but also ecological parameters. So a few data. We analyze the trophic level of the system, and you see the bi-monthly monitoring, and here we determine the trophic index TRIX, which integrate oxygen, chlorophyll, but also nitrogen and phosphorus level. And you see that the water frame, the unit 1, which is corresponding to the leisure boat sector, has a higher trophic level as compared to the other unit, to the unit 2. We also evaluate the trophic level by determining the solvent organic carbon and total counts, and you see that we have a clear linear correlation between these 2 parameters. However, we don't have a separation of the water body in the 2 water management units by this parameter. Then And I present some preliminary data from the Illumina sequencing. We analyzed the structure and composition of the prokaryotic community by 2 sets of primers, one for bacteria and the other for archaea. And here I present data with respect to campaign, winter, spring, and late summer, or management unit. It's really clear that you have a separation of the bacterial community with respect to the management unit, with the lower diversity in the management unit 1, while you have a temporal variation of the diversity of archaea. These are, of course, rarefaction curve with the rich estimator of the Howe index. As far as concerned composition, it's clear that in the management unit 1, you have higher concentration of actinobacteria here in green in all the seasons, which could be attributed to a higher pressure from the inland, while in the Station 3, which is located near the port entrance, you have a higher proportion of proteobacteria and cyanobacteria. Finally, as I told you, we have to manage with the bioremediation of and we perform a field scale demonstration of the most efficient bioremediation technology. As you know, oxygen, and several presentations talk on this today. Oxygen is crucial for degradation of hydrocarbon by bacteria, so we decide to measure oxygen level by oxygen saturation. And you see here the graph in the monthly monitoring. You have higher oxygen saturation in spring, then you have a decrease in the oxygen saturation in all the 4 stations. In September, we performed transect for measuring in more detail the oxygen concentration, since we have a low concentration in all the 4 stations. And we see that at station, at the port entrance, we have a wide fluctuation in the parameter, while in the inner part of the port, you have low concentration and constant low concentration of this parameter. Then the system recover, and you see that in the water management unit 2, you have a faster recovery of the system as compared to the leisure boat station. In this graph, I present data of heterotrophic bacteria and diesel degraders, and you see a bloom of heterotrophic bacteria in all the 4 stations in September, while you have the bloom in the outer station as regard diesel degraders, while you don't have any bloom in the inner part of the port. And we suppose this should be due to the low concentration in this area. Parallel, the higher concentration of hydrocarbon were found in the leisure boat station, so in the inner part of the port, and we found the higher concentration in September. You see the impact of the tourist season, and you see that after tourist season, you have the higher concentration of hydrocarbons in seawater in the leisure boats. Then you have a decrease, which is the recovery of the system, which correspond to a simultaneous increase in dissolved oxygen and diesel degraders. So you have an increase of both parameters. Based on all aforementioned data, we decided to perform a bioremediation treatment test in the Helcantaway port. This data will be present by Nicola Frigao, which is in the audience this afternoon in a poster. And we combine aeration treatment and sorption of hydrocarbons by sorbent material in order to improve the recovery of the oil from water. So these are our conclusions. We demonstrate that higher concentration of hydrocarbons are present in the leisure boat station after the touring season. Thus, bioremediation treatment cannot be considered a valuable technology for bioremediation of this management unit due to the low level of dissolved oxygen, which could cause eutrophication, of course. Oxygen level, from our analysis, is the main limiting factor to microbial degradation. So, coming to the aim of the work, which is transfer of tools to port authorities. In this case, the first measure that we suggest to our port authorities to modify water circulation in the leisure boat sector in order to improve oxygen level. This is an example of how we can manage. This, of course, is the consortium, and this work has been done by University of Cagliari and University of Florence, and thank you for your attention.
Actually, it's more a suggestion or something, because you show that you need more oxygen, and where you have leisure boats, it's a closed area, this red area there, and probably have more respiration by bacteria than oxygen that you can produce with algae. Well, if I am correct, at the port entrance, in certain moment of the year, you have more oxygen. So you may have it like, maybe it's not the case because there is a lot of land there, but if you have a second entrance, you cannot just grow algae there. It's not an open pond. But if you just have a second entrance, maybe you can bring more oxygen inside, like a secondary channel. You don't really need boats.
Of course, we are using
right?
these pores, which is a very small pores. I don't put the scale, but this is 100 meters. So it's something like a multi-well, not a very big system, which allows us to really understand how it's working. Of course, we see that plankton, zooplankton, which is and the other heterotrophic component of the plankton, have the same bloom with heterotrophic bacteria. So we think that the depletion in oxygen in the inner part of the pores was due to a combination of two factors. The higher trophic load, which correspond to a higher respiration, mainly of zooplankton, which is more concentrated in the inner part, while heterotrophic bacteria are almost the same in the entrance. But of course, here we have a problem of water circulation. We can use some mixer or some, because in this case, it's really impossible to open something.
Not even a channel. I don't know. You don't need boats to go through, right? Just a channel to have water circulation naturally.
Yes, but here you have
because if you
hotels, here you have the city.
Yeah. Because if you put a mixer, I mean, who is going to spend energy for bioremediation? Nobody. If you just put it. Yeah, that's so sorry.
We have some connection with the Ports Authority of all over Tunisia, and in Hammamet port, they did improvement of water circulation by mixer and the injection of seawater, which could be a yes.
No more questions. When we decide to have an IRA.
Okay.